Okay, so we want to talk about chapter 26, which is digestive functions. So, digestive functions are pretty straightforward. So, ingestion is when you eat or drink and you take something into your food or water or food or liquid into your mouth. Okay, motility is the movement of and the mixing of all of these things in the GI tract, and we'll talk about that. Secretion is how things get from like the walls of the organs or from the gallbladder or the liver, how they all get into the GI tract. Digestion is the breakdown. So we take that and we actually make it into two components. So mechanical digestion is physical breakdown. So when we chew it up and mix it, that's mechanical digestion. Chemical digestion is when we use an enzyme or something like hydrochloric acid and we chemically change the nature of the molecule. And of course, my numbering wouldn't. It's always got to be screwed up. Okay, 5 is absorption, so that's how things get from the GI tract into the bloodstream. And then 6 is elimination, how we get rid of the waste. So that, I think that makes sense. So 6 things. Okay, now the digestive system is basically a tube, and it starts at your mouth, and it goes all the way to the anus. They say in a cadaver, when everything is relaxed, the GI tract is about 30 feet long. There's several names for it, so they call it the GI tract, the digestive tract, and then sometimes they call it the alimentary canal. So it's the tube from the mouth to the anus, and on this side, it's labeled. Okay, now the accessory digestive organs are things that are going to help break it down. Okay, so they assist in the breakdown of food. So they either are going to, like the teeth and the tongue, they're going to help you chew it up, or they're going to make secretions that get dumped into it. So those accessory organs are listed on this side. So teeth, tongue, salivary glands, liver, gallbladder, pancreas. So they're going to dump in. So two basic subcomponents of the digestive system. Okay, now how is it organized histology wise? Okay, so we're going to start with the mucosa, which is where the food will contact. So I have that written down. We'll contact food. Okay, the epithelium is simple columnar. And if you remember from histology when we talked about epithelium, we're always talking about absorption and secretion. So that's the function of it. Okay, the lamina propria is immediately below the epithelium, and it's areolar connective tissue, and it's full of blood vessels and nerves. Okay, so that's how things will get absorbed. Now, something that's a little bit different is this layer. It's called the muscularis mucosa, and it's a thin layer of muscle that's underneath the lamina propria. I'll show you on the next slide. And what it's going to do is it's going to make the mucosa move. Okay, the submucosa is underneath of that, so I always think about subflooring. So this is the floor, this is the subfloor, so it's below it. The submucosa is made out of areolar tissue and dense irregular tissue. Now, what's special about it is it has some immune function. So it's got some cells in there. They're called mucosa-associated lymphatic tissue, or MALT. Okay, and then it has these clumps of MALT cells, which are called Peyer's patches. Now, the function of these is to protect your body and your blood from microbes getting in through your food. So they protect against the absorption of microbes. So it's kind of a line of defense. So if you think about your food isn't sterile, shoot, I have food isn't sterile. All right, so you need some protection because you don't want bacteria or other things that could be in the food to get into your GI tract. So that's the protection. Okay, now the muscularis, there's most of the time there are only 2 layers, an inner circular layer, okay, so it's going like this, it's lining the hole, and then an outer longitudinal layer that's going this way. Okay, so we have muscles going in 2 directions. There are some exceptions to that, and we'll talk about those. What they're going to do, those muscles, the smooth muscle, for the most part. Okay, so it's involuntary. Okay, so what they're there for is to move the food through the GI tract and to mix it. So the movement, the wave-like movement of things through the GI are called peristalsis, and I have a picture of that. And then there's also mixing. Okay, so I have some pictures of that. I'll show you what that looks like. Okay, so peristalsis is when we're pushing it through, and then mixing is when we're just going back and forth and we're mixing it up to try to break it down. Okay, the outer layer of the GI tract is the adventitia or the serosa. Okay, it's called the adventitia if it's outside the peritoneal cavity, and it's called the serosa if it's inside the peritoneal cavity. So that's just going to anchor the parts inside the cavity, inside the abdominal cavity. Okay, so here's a picture that's showing you mucosa. Okay, so that's the inner layer. Submucosa is underneath of that. The muscularis. So here is the circular muscle, and then here's longitudinal muscle. Okay, so you got two different layers. The myenteric nerve plexus is what's going to take nerves to that layer so you can control, so you can involuntarily control it. And then the serosa is the outermost layer. You'll look at this in lab on models, so you'll be able to see it and get your little hands on it. Okay, so now motility. So here's peristalsis. So peristalsis is kind of like, I think of it as like maybe a tube of toothpaste where you're squeezing and pushing. Okay, and then things relax and then you squeeze and you push some more. Okay, now the ball of food, what's going on with it's called a bolus. So this is the food after it's chewed and the saliva. So it's kind of like a ball. So it's called a bolus. Okay, so peristalsis is moving through. Okay, and then mixing is just going back and forth. So you can kind of see we're going this way and then we're going this way. So we're trying to have the bolus, what's in there, come in contact with how it breaks down. Okay, now this picture is trying to show you all of the layers and how absorption takes place. So if you think about structure and function, so the digestive substances we have microvilli. I'm going to show you this in another picture. Microvilli on the simple columnar epithelium. Okay, and then there's the basement membrane. So things can come through the GI tract, and they get absorbed. If they're big, like fat globs, they'll go into the lymph, and other smaller molecules will go into the capillaries. And we'll talk about this more. Okay, so now the serous membranes. Well, I don't know what's going on with some of this. Oh well. Okay, so the parietal peritoneum, it lines the inside of the abdominal wall. Okay, so it lines the cavity. The visceral peritoneum is going to touch the surface of the organs, and then the space between them is called the peritoneal cavity. Okay, so what's in the peritoneum and what's behind it? So intraperitoneal is the stomach, the small intestines, and part of the large intestines. What's retroperitoneal is the duodenum, which is part of the small intestines, the pancreas, some parts of the large intestine, and the rectum. Okay, and here's a learning strategy to help you remember where they are. And it's maybe kind of gross. So sad pucker. Okay, so peritonitis is when the peritoneum is inflamed, which that can be really bad. So if something leaks from the GI tract into the peritoneum, into that space, it's going to cause some pretty serious infection and inflammation. So we don't want anything to leak out because what you eat is not sterile. So we don't want not sterile things to get in that space. Okay, now the mesentery you'll look at with the cat or a pig. So the greater omentum is this fatty apron. Now you might remember this from doing the cat. It's the real fatty layer. So they call it the fatty apron. And in humans, it's where the fat occurs in our belly. Okay, so it insulates the abdominal organs. It stores extra fat. There's also a lot of lymphatic tissue in there. So lymphatic tissue is going to be important for immune function. Okay, now the lesser omentum actually attaches the stomach to the duodenum and to the liver, and it's easier to see it in lab. The falciform ligament attaches the liver to the anterior abdominal wall, which you'll see. The mesentery proper is the fan that keeps the intestines all together. And then the mesocolon is actually what attaches the large intestines to the back body wall. So you'll see it's easier, way easier to see in lab. Okay, now, how do we regulate digestive processes? So we have receptors in the mucosa and the submucosa. They're going to detect stretch or pressure. Okay, so when you feel full and it stretches, that's going to send a signal that, oh, they have enough, you have enough food, something's in there. Okay, the chemoreceptors in there detect hydrogen ions and fatty acids. So those will also stimulate secretions. Okay, now there's two kinds of reflexes going on. Okay, there are long reflexes, so they go all the way from the GI tract to the central nervous system, and then 3 cranial nerves send the information to the digestive organs. Okay, they'll coordinate motility and secretion. Okay, now there are these things called short reflexes, and they're just local. Okay, so they only involve the GI tract. So they're part of the enteric nervous system, so it's within the GI tract wall. We're also going to talk about 3 hormones. So gastrin, which is going to be released from your stomach, secretin, and cholecystokinin, which is released from the small intestines. So cholecystokinin is CCK. Okay, so the GI tract is divided into upper GI and lower GI. So first we'll talk about upper. So upper includes your mouth or your oral cavity. That's where chewing is going to take place. Chewing is also called mastication. So if you masticate something. Salivary glands, and I'll show you a picture in a second, those are going to release saliva when they know that food is in your mouth. Okay, and then I've already mentioned this, the bolus is the mass of food that's mixed with the saliva. Okay, the pharynx, we talked about with respiratory system, but it's going to be important in swallowing. The esophagus is going to be your pathway to the stomach, and it's posterior to the trachea. Okay, now the stomach is going to secrete gastric secretions, which they come from the epithelial cells, so hydrochloric acid, some digestive enzymes, and then this mucus that's called mucin. When that mixes with the bolus, that becomes chyme. Right? Okay, so here are the salivary glands. So the extrinsic salivary glands produce most of the saliva. So here's the parotid, where somebody gets mumps. It's the one that's infected. And then the duct that dumps into it. Submandibular, which that makes sense, right? It's under the mandible. And then sublingual, which is under the tongue. So those are pretty straightforward. Okay, now saliva, most is produced when we're eating, okay, and it's by the extrinsic glands. So it's kind of crazy. We make 1 to 1.5 liters a day. So it's a hypotonic solution, so it's mainly water, but what it does have in it is really important. So one of the enzymes is called amylase, and amylase digests starch. Okay, then we have lingual lipase. So if you look at lipase and lipid breakdown, so it starts breaking down triglycerides. Okay, mucus is in there. It makes it sticky. It helps lubricate. It has lysing enzyme, which is called lysozyme, and this will actually break down bacteria. Lyse bacteria, the cell walls of them. Okay, it has immunoglobulin A, so IgA, which keeps bacteria from growing, and it'll have electrolytes in there. Okay, so the function of saliva is it's going to keep the oral cavity structures clean, so like your teeth and your gums. So nowadays there are so many medications out there that cause dry mouth that people have a lack of saliva in their mouth, and then they end up with a lot of cavities or gum disease. So it's really important to have enough saliva in your mouth. Okay, the second thing that it does is it's going to moisten the food and make it stick together, and then it starts the chemical breakdown of the food. Okay, so salivation is controlled by some nuclei that are in the medulla and the pons. When you're resting and digesting, your mouth is nice and moist. Whenever you're scared and you have sympathetic stimulation, you end up with very thick saliva. Okay, so there are higher brain centers that can cause us to make our mouth water. So if you start thinking about food or smelling food or seeing food, it can cause you to salivate. Okay, then mastication is the last slide. So mastication is all about chewing. Okay, so the purpose of chewing is to make it easier to swallow the food. It's going to break it into little pieces. And what that does is it increases the surface area so that enzymes can get in and start digesting it. If it was one big clump like this, the enzymes would just have to pick away from the outside. But we want them to be able to come in from all sides and start breaking things down. Okay, the next thing that it does is it also softens and moistens the food. So all that makes it easier to swallow.